So yeah, today my title is Talk Geography and Genetics, Mirrors, Warped Mirrors, and Sampling. And this is work that I've been doing with my lab group over the years, and I especially want to highlight Desi Petkova, who is a statistics student who's graduated and gone on. Ben Peters, a postdoc in the lab, who's now gone on, leading the group with Max Planck. And Matthew Stevens, who was my postdoc advisor and has been a long-time collaborator for a lot of the work we've done here. So, great. So this is a Darwin Day event, and so I thought I'd start with an image of Darwin. This is 4 years before the origin of the species, and a pretty amazing photo. This look on his face, it looks like a man with an idea that needs to be told, right? And there's this great quote that he had about this own photograph. If I really have as bad an expression as my photograph gives me, how can I have one single friend that is surprising? Man, smart and with a sense of humor and humility. So one of the key things that took place between Darwin starting here in 1809 when he was born, the day that we celebrate with this event, and November 24th, 1859 at Murray's publishing house where The Origin of Species was published, was this trip, where he traveled around the world and saw many different ecosystems, saw variation. And it emphasizes to us that ecology and evolution are really illuminated and shaped by geography. In population biology, we ask how are populations connected by dispersal, and how does this affect their long-term demographic trajectories? In evolution, are there units within a species that are independently evolving, allopatric units, or loosely coupled, parapatric units? In conservation biology, we ask what are the geographical groupings we should manage for conservation? And in human biology and disease, we need to understand the genetics and geography of disease alleles to help use them in biomedicine. So the big challenge in our work, and the one that is the focus of my work, is moving from genetic diversity data, where we have lots of sequence reads that we line up to call genotypes. These are SNPs here, where you have two pieces of DNA, you line them up and you see a base pair difference, a single nucleotide polymorphism. Getting from this genetic data to some kind of understanding of the past to ecological and evolutionary processes. And the way that we do this jump from here to here is by using theoretical models and statistical inference. And so today I want to do that in the context of 3 vignettes. So the first is called the first law of geography, and genes mirror geography. We'll kind of review this nice result that I've been told you about. Then 2 asks, is the mirror warped? And then 3, for finding human disease variants, should you sample narrow or sample broad? So Walter Tobler, a geographer, has this line, everything is called the first law of geography. Everything is related to everything else, but near things are more related than distant things. Okay. Basic idea. And what I want to set up is showing results from the same study that Ben mentioned. It was from 2008 where we were able to get genetic data from a large collection of European individuals. And at the time, there was a lot of interest in using principal components analysis, a way of representing individuals in a small number of dimensions, oftentimes just 2 dimensions, and using these to correct for background population ancestry and environment in doing disease mapping studies. But there was little known of like what would happen when you used European data in this process with these large-scale SNP chip data SNP chips that were being developed at the time. So these tools for measuring SNP genotypes can measure, like the scale that we have, where the ability to measure 500,000 locations in an individual's genome at a time. Okay. That large because you're trying to map disease, which is distributed across the genome, so you need to look at a large number of places. So what would we see here? And especially with the models of an understanding by geneticists of how much variation is within and how little between, it's kind of unclear like what you would see in the scale of Europe. Okay. And these were our results. So these are these principal components axes. Each 2-letter code is an individual. The individuals are labeled by their grandparental ancestry. So they had questionnaires asking about their grandparental ancestry. And here we're showing just the individuals that have all of their grandparental ancestry agreeing on one place. Okay. So we're seeing sort of a deeper view of the population structure than from the looking today, where many individuals have grandparents from many locations. Okay, so two-letter codes, different individuals. These two-letter codes are ISO country codes. These are the median position of all individuals with the same codes. And then this is a legend here. And what I want you to do is compare to the geographic map here what we see from the genetic data with what we see in the geography. And what was amazing to us is how much the genetic variation is mirroring the geography of Europe. And so if you keep in mind on the left, this plot is made feeding into an algorithm just saying summarize the genetic data in 2 dimensions. You don't tell it anything about the latitude and longitude, where these individuals come from. You don't tell it anything about the grandparental labels. And so you superpose those labels onto this the pattern that emerges. Okay, and yeah, and so you see Spain, Portugal, France, Italy. Hopefully, you can sort of see going back and forth the two. There are some interesting outliers. But first, let me say that overall, this level of differentiation is very subtle. Where FST, a number that population geneticists use for measuring differentiation, varies between 0 and 1. 0 meaning there's no differentiation at all. 1 meaning that it's as large as it could possibly be. It's like those circles being completely non-overlapping. In this data set, it's 0.004. The proportion of variation explained, which is a number that's used in PCA analyses, is 0.3% and 0.15%. Again, very low numbers for, and suggesting still underneath all this, the circles are overlapping, to use Brian's imagery. Some of the interesting, well, I'll show, first label that the samples have less than 3, where the median is being drawn from less than 3 individuals. One of these is the SK point for Slovakia. Notice how out of place it is. This is because there was just 1 individual that had the label Slovakia. We've since gotten more individuals from Slovakia, and they actually do land up here. These 5 Italian individuals, we couldn't get any more information at the time about where their ancestry was from. We've since gotten access to Sardinian data, the island of Sardinia here in the Mediterranean, and Sardinians land right on top of it. Okay. Linguistic isolates. Hungarians speak a Finno-Ugric language, which is more related to languages, yes, Finnish, for example, and yet Hungarians in this sample cluster with their geographic neighbors. And then Romanian is a kind of linguistic isolate, that it's a Romance language spoken on the other side of a group of Slavic language speakers. And it looks like it shifts slightly towards the other Romance languages, but when you statistically look at this, it's not significant. This is kind of within the range of the wiggle that's happening in the genetic geographic map here. Okay, so if we just look at the genetic correlation versus distance, we see there is higher genetic correlation the closer 2 populations are, and that law is holding up here. Everything is related to everything else, but here things are more related than distant things. One thing I do like to do when I show this data is to stop and think about the underlying levels of differentiation and try to convey it in a more clear way. I gave you those numbers, FST is low, but few of you really have a sense of what FST means unless you study population genetics. And precisely for the reasons that Brian was mentioning, people can look at these plots and imagine deep levels of difference. One tool is to just grab 25 random SNPs out of 500,000, and let's plot the frequencies of the alleles across different regions of Europe. And this is what we see. So each of these lines is one location in the genome. The frequency, there are 2 types of alleles. We're calculating the frequency of that allele across 6 different regions of Europe. And you can see the frequencies are just fluctuating ever so slightly. Okay? So in general, the levels of differentiation are low. It's not that because we can that individuals from Spain are sort of one part of the graph and individuals from another country in another part of the graph means that there's deep genetic differences. In fact, there's just these subtle fluctuations. Maybe I'll just project. Is this okay? Can you still hear me? Brian was doing fine, and I'll try to do the same. So these are little wiggles, and these also include sampling error, because these are finite samples, so some of these frequencies are estimated with noise. So this is going to be more wiggle than actually exists in the underlying data. Another way to show this is with this sequence of plots. So This is what happens if we use just 100 loci, with 1,000, with 10,000, with 100,000. Okay. You need to squeeze the data. It's like aggregate the signal from hundreds of thousands of markers to start to see, oh, these individuals are a little more closely related to these other ones, and then start to see these relationships. Okay. This connects to a broader challenge, and one that I'm working on in my research, which is the challenge of conveying absolute levels of differentiation. So these are all examples of visualizations that population geneticists use, but few of them convey the absolute levels of differentiation, like sort of how much those circles overlap, to keep using that metaphor. They don't convey what are typical allele frequency patterns, like the ones I just showed you, of like how the allele frequencies are pretty constant with like little fluctuations. You don't get that from these visualizations. I saved that material, which I usually would input, for tomorrow's talk. For the public talk, I want to show some of those absolute levels of differentiation. So I'll show some new graphs that we have to try to do that task and tomorrow. Okay. And I'll skip along here. So, okay, so then, as I've shown this plot in different audiences, I sometimes get asked, well, you know, is that little gap there, is that the English Channel? You know, that's the Pyrenees Mountains, you know? Is that the Adriatic Sea? And I've always been really reluctant to try to like go that far, right? And part of that is before I ever did this study, I'd done work on PCA as a method. We have a paper that shows that you actually should get these geographic relationships when you have clean isolation by distance patterns, when that first law holds. But if your sampling is uneven, PCAs can get distorted in odd ways. So I worry that some of these gaps are just because of the uneven sampling. So to show that in an exaggerated way, I mentioned that we've gotten more Sardinian samples. If I stack this with 500 Sardinian samples, yes, they cluster with those other, 5 Italian individuals, but it also blows out the whole map. Okay? If I subsample the Sardinians and put about as many Sardinians as I have with other countries, I get something that looks more like the map with those individuals on top, right? So we have to be really careful, like where did the Pyrenees go? Where did the Adriatic go? It just got lost in this, So somehow the sampling really matters, and you have to be careful about that. Okay. So this connects to a larger question, which is how can we reveal what we call spatial heterogeneity in isolation by distance patterns? So we have an understanding that genetic similarity should decay with distance. What if it decays with distance at different rates in different places, right? And that's a question that is asked a lot in a subdiscipline that's sometimes called landscape genetics or geographical genetics or geogenetics or spatial genetics. It's used a lot in conservation genetics to try to understand patterns of gene flow along a landscape, to say, oh, we need to, it's really important that there's a lot of gene flow happening through this area, we might want to conserve that habitat. So it's a question that has been addressed with the development of many different tools. Tools for visualizing local allele frequency differentiation, such as Walmling, monokinase algorithm, local diff. Tools that are based on dimensionality reduction, like PCA, so doing maps of principal components, rotating principal components to a map. It's for Krusty's approach. Space mix is an approach that tries to represent the individuals like in a PCA, but also taking into account the geography. And then variants of a tool called structure that add in geography, so G-land test or construct is one of the most recent and interesting. So in this big space, different methods have advantages and disadvantages, and I'm going to tell you about the approach that we've developed. We call it EAMS for Estimated Effective Migration Surfaces, and we published it in 2016. This acronym EAMS is partly meant as a mnemonic to help you remember what we're doing. It's partly inspired by Ray and Charles Eames, who were furniture designers. And but before they were furniture designers, they were developing splints for soldiers in World War II, and they were bending plywood to the shape of the human body, right? And the complex shapes of the human body. And so what we're trying to do is bend these surfaces that you'll see to reflect the complex relationships of genetics and geography across the landscape and surface. One of these just went on display, by the way, at the Art Institute in Chicago. If you happen to be by, there's a new design exhibit, and I just have to be excited to see one of them directly. Yeah. Okay. So so let's introduce that a little more. So this is an example from elephants, forest and savanna elephants from Africa. So the green dots are forest elephants. The savanna elephants are in orange. This is a known ecotone here. That there's a divide in the habitat, and there isn't a lot of gene flow across it. That the forest elephants are kind of on one side of the ecotone, and savanna elephants are the other. And our method is going to analyze the genetic data, in this case 16 microsatellites, and try to produce a surface that informs us about the patterns of genetic relationships on the ground. So this is what we get when we run it on this data set, and we see this area here that has low relative effective migration that is telling us that there's not a lot of gene flow happening across the ecotone. And as funny as this shape is here, this kind of C shape, we can fit it. The blue here represents relatively high migration. So here, these are areas that have a high genetic similarity with each other, given their geographic distance. It seems that we have to model it by saying there's a lot of flow across here. And then in the forest area, we see kind of more average levels of effective migration, or even below average. And we think that's partly because these elephants are moving more on the savanna and less in the forest. Okay? You might notice that like this feature kind of extends upwards, even though there's some data from there. There is a kind of principle that you can learn about gene flow even if you didn't sample in an area, because the ancestors of the people you've sampled may have passed through there, and so if the gene flow is high or low, you actually can learn about it. But we caution, inferring too much outside of where there's sampling. And then also a thing that I'll highlight is we have this assumption of a kind of area that you're going to learn this landscape over, and that's kind of a construct. It's and a key kind of caveat of how you do this. So more precisely, we have this dense grid of populations that we imagine over the space. So now I'm getting under the hood of how this works. So this grid represents little populations that are exchanging migrants with one another along all of these edges. And we can parameterize such a model, an actual evolutionary model, with migration rates and population sizes. So now I'm coloring the edges by the migration rates that we might suppose exist across space. And if we had those parameters, that would imply pairwise coalescent times. That is, if we sample an individual from one place and an individual from another, and take DNA from each of them, how long would we have to go back until we find their common ancestor? If you give me a set of migration rates, population sizes, I can get an expected time. And if I have an expected time, then I can give you an expected amount of difference, because things that are more distantly related in time are going to be more genetically different on average. And so then it becomes a game of, well, let's try updating the migration rates and population sizes to make these expected pairwise differences match what we observe. Okay? And the way that that's done is using Wishart likelihood. I'll go a little quick here, it's a little more mathy, but we use a Wishart likelihood on the distance matrix, and then so we can calculate the likelihood of the data and update the parameters. And the way that we update those parameters is with a Markov-Monte chain approach, a Bayesian approach, and we use a Voronoi prior. So that's where this would be many parameters to fit at once, right? We have a parameter for each of these edges. So we simplify it by saying there are big areas where the migration rates are the same. So just learn how many areas there are and what migration rate is associated with the area and where those areas are. And so that can be formulated as thinking about having a tessellation, a set of tiles over the space, where you want to move the tiles around until you find a good fit. And in practice, practice, what we do is we're sampling these different tilings and then averaging over for every location what the posterior migration rates are at that location and the diversity rates. Now this migration here, we have to be careful about how we interpret it when it comes out, because it's an effective migration rate. So those of you who study population genetics will be familiar with like migration rates times n, what's called NN, and that's kind of what we're inferring here. So we're pragmatically to show it. This is a population where there's a trough of migration, where there's lower migration on where these dots are, and higher migration where the squares are. And if we run this in Eames, we get back a map that has low migration in the middle and high migration on the sides. That all makes sense. Okay, it seems like it's doing what it should. And here, I'm going to argue it's still doing what it should, but for a different reason. So here, in this middle area, we're saying the population size is low. The migration rate is constant. And we still get a map like this. Okay? If you literally, all you care about is migration rates, then yeah, you could say this is doing things wrong. But if you care about gene flow, like how connected are these populations, like what matters for evolution, then this is an interesting feature. The fact that the population size is lower here means that genes will move less often between these two places. And so there will be an elevated amount of genetic distance per geographic distance as we look left, right, across this. Okay. So we have to be careful as we interpret these not to just interpret it simply as being the outcome of migration. It's migration and population size. And that's why we call this a model-based visualization. It's a way that we use a model to reveal structure in the data, but not really for precise parameter inference. Okay? We're not, because we're making choices about the grid size, how many populations are on the grid. There's a lot of aspects there that we don't really know the actual answers, right? Of how dense the grid is. But by putting this fiction out there, that there is a model, and then asking what migration rates would fit that model, it allows us to see structure in the data that we otherwise wouldn't be able to. This is an example where it's been applied to these blunt-nosed leopard lizards, an endangered species in California. And so these lizards live on the southern end of the Central Valley in California. And these are samples where they've taken the colored dots. The colored dots are related to the results you get when you run structure on this data. And you'll see here that there's this Tulare Lake that they've drawn on the map, which may frustrate some of you if you're from California, because you're like, wait a minute, there's no lake in the middle of the Central Valley. And you're right. This is Tulare Lake in Google Maps today. Okay? This lake got drained and converted to farm pasture around 100 years ago or so. And so now the lizards can move across this, it would seem, quite easily. Okay. The structure, so this is the structure now. So this is what we get with Eames. And what's fascinating about it is, first off, you see the largest sort of feature that's causing low migration is down here, where there are known mountains that exist. But what surprised these researchers was to see this low migration feature here. But it makes sense when you understand that there was a historical lake there, and this is where the Kern River used to flow into the lake. This ancestral feature of the habitat is still structuring the genetic variation on the ground. Okay? That's what the title, Persistence of Historical Population Structure in an Endangered Species. So as you think about conservation units, you might not want to just say, oh, this is Central Valley, near the old Tulare Lake, and no big deal, they're all the same. Like here you understand, okay, yeah, there's a little more differentiation across there than you would expect otherwise. Okay, so going back to the human data, where people, were asking, well, are there wrinkles, is there somehow are the Pyrenees distorting the map somehow? We can wonder, will the PCA on human data give us something that is it so cleanly geographical that Eames will infer a flat map with no local changes in effective migrations? Or will it show something that has these wrinkles? And also, how will it compare to PCA as a mechanism for visualizing data? Because that's one of the main tools that people use for spatial data visualization. Okay. the result from applying it to a very large data set that Ben built by collating together 26 different studies. So each of these dots represent individuals that are sampled from different populations around the world. There are questions like caveats to be had about how individuals are sampled in any one location, about how they're registered to a geographic location. So we have to be cautious about that. We're trying to be somewhat optimistic that at this scale, exactly where you place individuals hopefully shouldn't have too much of a impact. But I want to contrast like what you see in a PCA analysis of the same data, which actually this is probably the first time any of you have seen a PCA of the whole globe, where you see Africa and then kind of a gradient going into Europe and then gradients going into Siberia, China, East Asia, and this data set is just Africa and Eurasia. Yeah, if it were we had the Americas in here, we'd keep going. And then interesting to see Madagascar, which was settled largely by Southeast Asian individuals from Indonesia, and then has had a mixture with African populations. And so it sort of sticks here between Southeast Asia and Africa. It's like a Sardinia in another way. Okay, so you see this broad scale structure, but what I highlight is in the Eames version of visualizing the data, we're doing something new from here. We're putting these dashed lines where the posterior distribution has more certainty. Okay? Because sometimes you can have a flash of orange that might not be that confident, because there's like in any inference there's uncertainty. And so these marked ones where there's more certainty that the feature is real. And so notice how you can see the Mediterranean, or the Red Sea, the Black Sea, the Caucasus Mountains, the Caspian Sea. And try to see those features in the PCA analysis. You would have to dig through many lower PCs, looking at the geographic map, looking at the PCs to try to see those distortions, and even then you'd be wondering about whether it was just sampling effects. So we're pretty excited about this as a way of seeing a lot of the features that exist in a data set in one go. If we zoom in on Africa, this feature here that runs across the Sahara and then wraps down, I wasn't sure at first why it would just not stop at the Sahara and then that there wouldn't be connectivity through here. And then this linguistic map really kind of cleared it up for you. This is where historically, and today, the boundaries of where speakers of Niger-Congo languages, the A and B groups, are distributed relative to Nilo-Saharan and Afro-Asiatic speakers. So that kind of gives some more support that this is a kind of meaningful area where human variation changes over remarkably for a given geographic distance. You see a feature here that's sort of in the Congo River Basin that's also kind of around the areas where the two subgroups of Niger-Congo are spoken. This feature around the Kalahari Desert where there are many Khoisan speakers, and these are sort of the click language speakers, and many groups in Africa that are still sort of practicing pastoral and other practices. So we have this a view that kind of nicely aligns with some of the linguistic and physical geography that's known. And actually, I use this example too because in a lot of ways we're one of the reasons to study humans in depth is because we know things about them, like language families. So if we can show that a method works, and we can understand that it's working and get kind of validation, it gives us more confidence when we go and apply it to other species for conservation uses, etc. This is looking at Eurasia, and here, I was already talking about this. Sort of, you see that the effects of these seas is where the Caucasus Mountains are, where the Ural Mountains are, the Tibetan Plateau, the Himalayas kind of coming through here, areas where the Gobi Desert and others, some of the mountain ranges that are in northern Siberia come through. A lot of features that sort of align with physical geography. Okay, now, so the answer is that yes, the mirror is warped and it's affected by physical geography in many cases. Now we bring it to the third part, which is to sample broad or narrow. And this is new, unpublished work. A powerful new trend in human disease genetics is the use of biobanks. So this is collecting a large number of individuals with lots of phenotype data, including electronic health records, and using these to study the genetic basis of biomedical traits. So the UK Biobank is the most powerful to date. It has 500,000 individuals sampled in the UK. There's a similar project in Japan, in Finland, in China, and the US has started a project called All of Us that is currently enrolling individuals. And there's a question of, is this a problem? How big of a problem is this to be sampling some areas incredibly deeply and missing out on many other areas, right? As we think about that, we need to think about how, as we're doing biomedical disease genetics, we're trying to find mutations that, one of the goals is to find mutations of large effect, because mutations of large effect on a trait, they give us a clue about the core pathways that are involved in that trait. Many of the variants that have small effects are just sort of like not directly related to the phenotype in question. And so this is a plot from results of studies of human stature, human height. This is frequency on the x-axis and effect size on the y-axis. And most of the variants that are common in a population have very subtle effects, and the things that have large effects on height tend to be very rare. That's actually an outcome of evolutionary theory, that if a trait has any kind of stabilizing selection on it, there won't be large effect variants that exist in the common variant frequency. So we care in a lot of ways about rare variants. Rare variants tend to be geographically localized. So this is a cartoon where each of these circles represents a population, these edges represent migration, and the dot is, okay, so this is populations, and this is one locus, another locus. The blue is a new point mutation that occurs. And then it starts to spread in its local population. And then it starts spreading to neighboring populations until it's more widely dispersed. Right? So these rare variants may be geographically localized. A challenge is that human disease genetics right now is this huge bias. Okay? And Carlos Bustamante and colleagues did a wonderful job first drawing attention to this in a big way in 2011 with this article, Genomics for the World. And Alicia Martin and others have more recently cataloged this by year, the number of individuals who involved in GWAS studies, the major medical genetics form of study, and then with stacked bar charts showing their representation of different ancestries. And it's been increasing immensely. And down here you can see just as a fraction, and the proportion of European ancestry is basically really high and stayed really constant. There's a slight excess increase in East Asian ancestry that's been included. There's more studies happening in other parts of the world, other ancestries, but in general, it's still really poor. This is an example from the UK Biobank, where they use ethnic groups that are based on self-reported backgrounds, and 94% of it is white British in the terms that they use in the study, with much lower representation of other ancestry groups. So what are the consequences of this? How does sampling in a few parts of the world, with mainly single ancestry types, impact the results and our chances for success? So should we sample broad or narrow? This is a cartoon sketch of the problem. Here, this is representing broad sampling, where we sample broadly. Here, narrow sampling, and then this is a schema of a map with latitude and longitude. And these dots represent the rare variants that we want to discover, these things that are possibly very informative about disease biology. And in the broad sampling case, we'll discover more of them, you might think, because we're sampling broadly. In the narrow sampling, we might get, well, maybe we're actually going to have more power because we're going to get everything that's right underneath us, right? So it's sort of this challenge of like, if I spread it out too wide, then I won't get anything rare and I'll miss out. But then maybe I'm going to get more because there's more of them out there. Versus sampling deeply, I'll be sure to get what's there, but if there aren't many there, then maybe I won't get much, right? So this is a place where you can't just logically reason it out. It's a place for evolutionary theory to help with the design of medical studies. And my postdoc, Dan Rice, has worked out the set of partial differential equations that allow you to work out what combination for each frequency level that you would see that an allele could be at, from like very rare to very common, how many mutations you would find as a function of the spatial dispersion. And this, the equation for this, for those who are hardcore PopGen geeks, this actually is kind of cool because it has the same shape as what you get for the site frequency spectrum under negative selection, but where you have different effective parameters that are controlling how steep this negative selection sort of effect is. Okay, but more pragmatically speaking, what do the results mean? So the blue line here is a very broad sampling, and what you get is discovery of many alleles that are very, but they're going to be very rare. If you do narrow sampling, you get more that are at higher frequencies. And so, yeah, this is in the broad sampling you get more, but you get many that are rare. In the narrow sampling, you get ones that are at higher frequencies because you've sampled a lot of them in that one place. And so what's the trade-off in terms of just discovering new variants, right? Like just in terms of finding more alleles. For just discovery of rare variants, what you find is that if you do so relative to just sampling in one location, the broader you make the sample, the more you have to sample. Okay? So there's this dilution effect that's causing you to miss more variants as you go wider, and so it's actually better to just stay narrow. Okay? There's a trade-off to doing this, though, because if I discover that just the variants in my population, and I don't just want to understand disease biology, like in the sense of like, if I learned that a variant is very damaging for people in this one place, okay, there are few people who carry it, that's telling me there's some way of like perturbing the system of like, there's a lever I can push that will affect this disease. And that's really helpful for like thinking about drug design. Okay? But if I'm trying to do preventative medicine, like predict, okay, I can do well for the people right in my local zone, but I'm going to do poor as I go further out. And you can do the math on this as well, and this is showing your relative ability to explain heritability, which is directly related to how well you can predict, as a function of distance from where your study took place, and as a function of how broad your sampling was. And the broader the sampling, the better you do at explaining the heritability of individuals who are farther away. If you're only interested in individuals who are closer, then yeah, doing narrow sampling helps. But this is a key issue. Okay. So so there's this trade-off. It depends what your goals are. What you mean by precision medicine. Is precision medicine, like, is the goal of the All of Us program to get better at trying to predict disease outcomes for individuals, or is it to help us discover drug targets, basic biology that then we can like build therapeutics around that would hopefully work for everybody because of how similar humans are. So those are the three vignettes. Just to summarize, the first part, yes, is published. It's a result for Europe. You see similar results in other parts of the world. Ancient DNA studies actually suggest this pattern is relatively young. If you go to the Bronze Age in Europe, you actually don't see it being such a clean geographical story. It's more that there were the model now is that there were sort of hunter-gatherer people that mix with farmer people, that mix with pastoralist people, and then the mixing that's taken place since the Bronze Age has led to this geographical relationships. This human generation is lower than most species, that FST being so low. So if you work in another species, don't get discouraged, like, oh my gosh, I need to have 500,000 SNPs to see the variation in my species. Because in most natural species, the levels of differentiation are higher, and so you actually can get away with less data. Okay. The part about is the mirror warped? That's the first methods paper is published in 2016. We have one extension paper that's already published, and we have another one on the way. Basically, I would convey that this is steps towards a dream. These maps are one map, even though migration changed through time. But we'd like to get to where we can build multiple maps, and this was just our first step. already a useful step. It has a lot of applications, but we're still just dreaming of how we could get maps changing through time, directional migration as well. Okay, and then in terms of sample narrow broadly, this is ongoing work, and we're trying to characterize these trade-offs more precisely. In closing, I want to add, and I've been doing this for a while now, a few words on genetics and society, and I'm just like thrilled that I'm getting to meet Brian in person after reading about his work, because he's one of the leaders in this area. But we're in a difficult time, and of where white nationalists, other groups with strong ideological motivations are looking at genetic data and interpreting it to strengthen their ideologies. So these are this group that's pouring milk over themselves in front of an art installation in New York. You say, why are they doing that? And it's because there's a mutation in humans that called, well, there are several called lactase persistence alleles. And one of them is found at higher frequency in Northern Europe. And it was the first one to be discovered. So in many evolution classes, it's taught, oh, there's this mutation that's in high frequency in Northern Europe. It confers the ability to drink milk as an adult. And it seems to have been increasing in frequency because it helps human health. So it's the kind of story that we as biologists tell in intro bio classes to like help pre-meds get excited about, wow, that evolution matters for humans. Maybe I should think about that. But at the same time, right, there are kids who are, reading it the wrong way, okay? And the biology is really working against them because the mutations actually, there are several mutations in different parts of the world. It's not just one. The same mutation that is found in Northern Europe is actually backflowed into Africa because there is that much mixing between the continents' boundaries that like are not, right? Even those, as I showed those troughs in migration, they're very permeable. It's just meaning the migration is lower across the Mediterranean. It doesn't mean people aren't moving across it. So everything works against them, but they're absorbing what we do and racializing it. And so we have to be more sensitive and do the kind of hard work that Brian's helping lead the way on. And I'll just share some of the things we're doing at U Chicago in case it might be useful to think about thinking about here, and I'd love to hear about practices that folks might be doing here. One of them is that many universities have an ethics of research course for biologists, and we've actually said that should include a section on genetics and society. And so we have actually 2 sessions now because we found we needed to. One on history of eugenics in the US and genetic engineering, helping people to talk about the ethics of things like CRISPR. And then another on genetics, race, and discrimination in biomedicine. We've started in our departmental seminar series inviting social scientists and humanities scholars, which was not something we were doing before, and that's been really productive to spark conversations and show our students that these issues matter and learn for all of us. We've added some discussions into relevant courses like our human genetic variation and disease course. And then we've had some journal clubs, our departmental journal clubs, where we've, instead of talking about a scientific paper, we've talked about things like the Genetic Information Non-Discrimination Act, or genetic data ownership. And I'll plug this one little Twitter account that I've started to like repost articles about genetic discrimination. And in particular, I want to highlight Anya Prince, who's, well, just coming up in a minute, but she's beyond, I think she's going to talk about genetics and law enforcement and those kind of issues. She's also Today's Project person. She's a leading scholar on genetic discrimination generally. So with that, I need to get off the stage, I think. I want to thank all the folks that were involved in this work and my funding sources, and Brad McRae, who helped develop some of the work that was underlying these. So thanks.
Maybe 1 or 2 questions if we have any scientific mapping people who want to engage with this. We will take a break then for just about 5 minutes, 7 minutes. There's coffee and cookies out in the lobby, so feel free to help yourself. Anybody got any questions for John? What?
Yeah, different groups have called these things different, given them different designations for subspecies, and the FSTs in that example are really high. They're like 0.6, and they're detectable. That boundary is detectable with just 16 microsatellites. So yeah, the two sides are very differentiated, and that's partly why we started there. It's like an easy test case. Yeah.
Yeah, you talk about lactose tolerance and intolerance and its geographic relationships. I was thinking about another example, maybe even more prominent, and that is high altitude adaptations in places like Quebec, which I understand are at least partially genetically based. You showed this map indicating this swath, this region of lower migration around the Himalayas. Is there any association, relationship between those two phenomena?
With so I specifically about migration in the Himalaya and the adaptation to high altitude. I don't know enough to make a precise statement on that, but one of the basic, if we use like evolutionary principles, local adaptation requires time, and also that there's kind of a balance between if there's too much migration with outside areas, then you kind of swamp and dilute the locally adapted alleles. So